Yes, I thought I did.
I think it's already up on the Discord.
Yeah, I thought I did put both of them there.
Paper 3 and 4.
Who was asking before? I didn't get the name of the person.
Sorry, it's Alessandra. Okay, my bad. I'll check it then.
Just check it. Yeah, I always post it a little over a week before so that you have the time to read it and work on it. Okay, I think we're going to begin now. Good morning, everybody. We are talking about mainly the topic is secretion systems now. In the beginning of this chapter, we were talking about how nutrition gets across the membrane into the cell. Now we are talking the reverse process where the cells secrete proteins out. Not only out, but some of the proteins have to be embedded in the cytoplasmic membrane. Some have to be transported up to the periplasm in the gram-negative bacteria, and some will have to be transported to the outer membrane in your gram-negative bacteria. So definitely protein secretion is a major topic in bacterial physiology. And as we go into more and more systems that are being discovered and being characterized, and similarities and differences are being noted, you will see that this, what I am presenting, is not all current. There is more to it than. So I just wanted to start you and warm you up to this topic of how many secretion systems that we have discovered in different bacteria to deliver either proteins and sometimes even DNA across the membranes in the environment or into another cell. Okay. So with that, we will talk about. We talked about the systems that operate in the cytoplasmic or plasma membrane, the ABC system, the TAT system, and all. Now we are looking at some more systems which we categorize in type 1 through 6 or up to 8 now. I saw in your new book they've gone up to type 8 also, and this is mainly for describing different types of transport systems across the outer membrane, and which of the transport system in the cytoplasmic membrane work with which of the transport system in the outer membrane. So there is a lot of information here. What I want to tell you is that don't get all worried that you have to memorize all of this. The idea is to be able to look at a picture like one that I'm showing right now and be able to categorize it as to which type it is and what are some of the components of it and what is an example of it. The most important thing is in which bacterium and what was being secreted should be an example that you should remember. So example for type 1, you should know an example or two. So I will ask questions about which, to give an example where this has been discovered in which bacterium or what is being secreted out. So just remembering that is important, not every component of it, and be able to differentiate between them. Okay, so in this slide, we are looking at, remember, the type 1, which is almost like the ABC system, where you have a pre-protein that has an N-terminal signal sequence and also has a C-terminal secretion sequence. So two of those are there. And then the ABC exporter consists of the cytoplasmic membrane, ABC. Then there is the membrane fusion protein that connects the two membranes, and then the outer membrane protein is there. And this whole channel allows this protein to be secreted out. Okay, and of course this is dependent on the ABC system where ATP binding cassette is used or ATP is being hydrolyzed, and that energy allows the secretion of that protein there. We look at type 2 now. Let me put it on here. So make it better. I need to know, yes, we are. Last time I forgot to record the lecture. Please remind me in the beginning. Sometimes I forget to turn it on, and then So last time's lecture was not, it will not be available because I did not record it. Anyway, this is the type 1 we just talked about. So we talk about the type 2 now. And now this type 2 secretion system is using the sex system again, the sex system, the secretory pathway in the plasma membrane. And this protein comes out, it gets folded, and then it is channeled through a GSPD secretin. So this channel is formed by another system, which this will capitalize on and be passed through as a folded protein now. So this GSP system, which is a direct connection again, where it uses, similar to your sex system, it is using the hydrolysis of NTP, and then a direct channel is formed. So there is alternate ways in the type 2 where it could go through the sex system or it could go through the GSP system. So similarity there. So here is an example of the type 2 where you're looking at the pilus formation. You know that the pilus is produced by bacteria that can be extended out. So in this model which you're looking at is your proteins are being secreted up to the periplasmic space where they get folded, and then they go into the GSP system where the channel has already been formed. Actually, these proteins are now that are folded now, that are being polymerized now onto the pilus. We call it the pseudopilus, and until it comes out, it will be the one that extends out of the cell. So actually, these proteins are being assembled on this the pilus, which will now extend. As more and more polymerization occurs, the pilus will extend out of the cell. And so there is the potential of depolymerization also, so that the pilus can be retracted. And we'll talk about why the pilus needs to be retracted when we talk about conjugation. So this pilus extension occurs by adding the subunits of the pilus that come through the sex system, and they are being assembled, and they make the pilus grow longer here. So this is just another model of it as to how type 2 secretion system is used for pilus formation. We come to the type 3 secretion system, and this is a very important one. Sometimes it is abbreviated as TTSS or T3SS, whichever. Type 3 secretion system is a very important secretion system, usually used by many gram-negative pathogenic bacteria, because they are capable. This system is capable of delivering the effector proteins directly into the host cell, into a host eukaryotic cell or so. So this is a very important system that has been looked at, and this has been looked at in the case of delivery of some toxins or proteins in plague, which is Yersinia pestis is the cause of plague. You're familiar with that. Typhoid is Salmonella typhi, and dysentery, this bacterial dysentery is by Shigella. All three of these are gram-negative bacteria, and they all seem to use the type 3 secretion system to deliver the toxin from the pathogen directly into the host by making a contact using this type 3 secretion system. So as such, it's been seen that this the model shows that there are about 20 proteins, which will be 20 structural proteins. There will be the effector protein that will be delivered, and there will be some chaperone proteins also involved in this whole system of delivery in the type 3 secretion system. So this is an idea that we have the type 3 going through and making a pore or attaching to the host cell and delivering. So here is the comparison of 2 system versus type 3 system, and we'll talk about the type 4 also very soon. Type 3 secretion system, as we said, secretes virulence factors from gram-negative bacteria from the cytoplasm across the plasma membrane, through the outer membrane, and into the host cell. And this secretion machinery has been modeled as the syringe shape, and this allows a translocation channel to be formed between 3 membrane systems. So the best studied example of the type 3 system is the Yersinia. The Yersinia pestis that causes plague delivers the YOP proteins straight into the host. Now there is a the figure is incorrect. The new book, the new edition of your book has corrected this already, where this channel that is formed in the host cytoplasmic membrane is composed of the YOP B and D. So if you look at it, the YOP B and D form a channel in the plasma membrane of the host. Okay, so if you're looking at it, the system starts in the cytoplasm of the bacteria. This is the bacterial cytoplasm. This is the cytoplasmic membrane of the bacterium. There is the periplasm of the gram-negative bacterium, Yersinia being a gram-negative bacterium. There is an outer membrane that is being shown. So there is a channel that is made through and through using this YSC secretion system. YSC is definitely all the proteins that are needed to assemble this type 3 protein injection system. We call it the injection system because it makes like an injectosome directly from the membrane of the pathogen into the membrane of your host. Okay? So the YSC proteins are forming the channel and making the injectosome, but what is delivered out is definitely, I'll show you in the next figure. But anyway, here two systems are helping the protein that is being produced, the toxin or whatever, the protein that is going to be harmful to the host is being guided by the SYC and the RNA, both of them, and they are being channeled into this whole injectosome. So here, another thing that I should mention, that there is an analogy with flagella. So when the flagella synthesis, remember we talked about how flagella is synthesized using the basal body, and eventually the filament is assembled outside of the cell. So this type 3 very much is similar, and it seems like there is a lot of analogy between the flagella synthesis and the flagella apparatus to this type 3 YOP proteins, as we call them. Okay, so if you look at the next slide or so, you can see that this system has a, you remember in the flagella I told you there is a cap protein. If this cap protein is opened up or it's not there, then whatever the flagella subunits are, that they will be excreted out. So in this also, we are looking at in the Yersinia, the channel is formed between the inner and the outer membrane, and there is a cap there. But when it comes near to the host cell, where it is going to be, the cap has to be opened up, and a channel is made through the plasma membrane of the host cell here by the B and the D YOP proteins, and the proteins that are delivered into the host cells have been labeled as YOP E, H, M, and O. So these are different proteins that have different functions, and as you look at it, the YOP E has the function to depolymerize the actin in the host cell. YOP H has dephosphorylation action on the signal transduction proteins, and your YOP O is the phosphorylation. So phosphorylation and dephosphorylation mechanisms are controlled by the YOP O and H proteins, while the function for YOP M is not known, but we know that all these four proteins can be delivered once there is contact between the host and the bacterium. Then the channel opens up, and the YOP B and D make the channel first, and then these proteins are then delivered into the host. Any questions on this one? Okay, so now we're going to show you the similarities. As such, if you draw the models of the Yersinia transport system, which is a type 3 system, and here is the flagellum. If you remember, the flagellum also has these MS ring and then the L ring in the outer membrane. So similar structure has been modeled for the type 3 secretion system, where ATP is used for channeling the subunits of the flagellum, not the flagellum, but transporting the proteins in the Yersinia system, whilst in the flagellar rotation, there is an ATPase involved, but also that this rotation of the flagellum, of course, is uses the proton motive force. So that is when the proton motive force occurs, then only the flagellum will rotate, right? But the structural similarities are very much there. We need to compare the two and see that that secretion system is very close to what the flagellum is like. Now, the type 3 secretion system is the genes for all of this is in a plasmid. So I'm showing you this plasmid, which is a 69.5 kilobase pair plasmid, and it has all the system for secretion, intracellular delivery, effectors and chaperone proteins are all coded on this plasmid. So definitely a very important plasmid found in the Yersinia, which is giving its virulence factor to be able to make the type 3 secretion system. And this type 3 secretion system will deliver the proteins into the host and cause the harm there. Okay. So in this plasmid, you can also see the other genes that are kind of interesting to find that they have been located in this plasmid and have been assembled in this plasmid, such as the bacterial adherence gene, arsenic resistance gene is also in here. So definitely it is a very interesting plasmid that has been studied in terms of Yersinia that allows Yersinia to produce the type 3 secretion system and allow it to deliver its toxins into the host. Another kind of from another book or from another paper, we found this also. So I give you a lot of different diagrams for the same idea. So if you don't understand one of them, you will get more information from the other one. So here is the comparison between the flagellar biogenesis. That means how is the flagellum created by the extension of the filament, and how these proteins go through the flagellar hook, the basal body into the hook and into the filament, and how these subunits of the flagella are actually incorporated at the tip of the flagellum. We've talked about that, right? And here is a comparison of the type 3 secretion system, which is, of course, the example of your Yersinia. YOP is always the Yersinia one. And this is a nice comparison also. So different figures from different sources telling you the same idea, that the flagella and the type 3 secretion system are quite similar in their in how they are laid in the gram-negative bacteria. Now, by using a scanning tunnel electron microscopy, this injectosome, as we call it, the injectosome, because it injects the toxin straight from the pathogen into the host. So by this, you can see that there is a tip of the injectosome that actually locks into the translocator pore on the target cell. So if you look at this scanning electron microscopy pictures, you can see that this injectosome tip locks into the translocator pore. We call it the translocator pore, which is also created by the genes in the Yersinia only. And these proteins create the pore, and then the contact is made direct, and the YOP proteins are released into the host cell. And here is what we had shown you as to how the flagellar components, each of the units synthesized in the cytoplasm of the cell, get through the basal body, through the hook, and are added into the flagellar extension the filament extension. And of course, there is the cap protein, which allows it to stay within the formation of the flagellar filament, rather than if this cap protein was not there, this flagellant protein would be released in the environment. So here is a comparison and some examples of type 1, 2, and 3 secretion systems for you to, now that we've come up to 1, 2, 3, let's look at them. So type 1 the example of a type 1 is hemolysins that are secreted by E. coli, which is the toxic one. EHEC strain of E. coli produces a hemolysin, and that is an example of the type 1, which is being shown here. Then type 2, the example is Klebsiella oxytoca, where the type 2 system is being used to secrete an enzyme called pulinase, which is secreted into the environment. Whilst your type 3 secretion system, which is the Yersinia pest, is the best example of it, showing how the complete channel is produced as an injectosome, and the protein is then released into the host directly. So giving you some more comparisons and giving you examples of these type 1, 2, and 3 secretion systems. We move on to type 4 protein secretion pathway. This is a unique pathway in a way because it not only secretes proteins, but it transfers DNA during conjugation. So when we talk about conjugation, we know DNA from one cell, which is the donor cell, it will go into the recipient cell, and this DNA has to be transferred across the membranes from one cell to the other. So it has to cross quite a few channels of the plasma membrane of the donor through the outer membrane of the donor into the outer membrane of the recipient and the plasma membrane. So all these membranes have to be crossed if the DNA has to be transferred during conjugation. So type 4 system is unique, that is used for DNA transfer during conjugation. Type 4 system also forms a syringe-like structure, and it has been found in both gram-positive and gram-negative. So it is not only limited to gram-negative, it is also found in gram-positive bacteria also. So let's look at some examples of your type 4. Here we've seen the type 4 injection system being used by Legionella pneumophilia, Legionella pneumophilia that causes legionellosis, that's a disease, and Helicobacter pylori, which is also a pathogen that is found in the stomach of some individuals, and eventually leading to I would say cancers of the stomach also. And then also these two, Legionella and Helicobacter, transfer their toxins through this injection system, whilst the Agrobacterium example, Agrobacterium tumefaciens, we will look at, is the one where the DNA is transported from the bacterium into the plant through the system where the DNA will be transported. The TI plasmid will be transported across from the Agrobacterium into the plant. Okay, so definitely there will be some similarities and some differences. So if you're looking at what's happening here, so it is being modeled into this picture for you. The SEC system operates in the cytoplasmic membrane. The protein comes into the periplasm, unfolded, it is folded, and then it can be channeled through this system, which is a type 4 secretion system. Whilst when the DNA is being transferred through this system, it does not use the sex system. It actually has to go through the channel or the injectosome that is created for both the membranes to be connected so that the DNA can travel through it and it will go into the host cell. We'll look at it a little closely in a little bit. So definitely type 4 uses sex system, but it does have an injectosome also in some of them. So here is a model of it, again, an easier and a simpler picture for you to see. This secretion system is being used for Bordetella pertussis toxin, which causes whooping cough, this disease, pertussis. So the secretion system 4 is being used for that. And you can see that the sex system is being used to transport the protein into the periplasm, and then using the type 4 secretion system to get into. Now, if this secretion system, of course, will deliver the Bordetella toxin directly into the host eukaryote through the eukaryotic cell membrane. Whilst when this system is being used for the DNA does not go through the sex system. It goes directly into the injectosome, and it is being delivered into another bacterium, is what we will. Or if it is the TI plasmid, then it will be going from the bacterium into the plant. Any questions now on all these figures that we are kind of discussing? I'll tell you, there are hundreds of papers all on secretion systems, and we really cannot, work on each one of them. We will have 2 samplings of those in the 2 review papers that you're going to be delivering next week. But there is so much literature in these secretion systems and how they are expanding as years go by. We have more and more secretion systems being discovered in different bacteria, and we are trying to draw parallels between them to say where can we place them in the, or already that we have categorized them into these types, where do they match up and where should we place them. So this is a very, I would say, a very open research that is continuing and expanding as we learn more and more. We move on to the type 5, which one of your papers will be discussing. Type 5 protein secretion pathway. It says it's the most recently discovered, but that's not a true statement because there are now 6, 7, and 8 also that have been discovered. So definitely one of the newer protein secretion system, which was fascinating them. And this relies again on the SEC pathway across the plasma membrane. But then across the outer membrane, a channel is formed by the protein itself. So we call it an autotransporter. Autotransporters are proteins that are moved from the cytoplasm into the periplasmic space through the SEC system. And then from the periplasmic space, it actually makes its own way out. So part of the protein becomes the channel, and part of the protein will be secreted out. And that's why the word autotransporter is given to such proteins. So here is the figure or the model for it. So your protein is coming through the SEC system. It is in the periplasm, and there are 2 ways that it will be transported out. One, by using the autotransporter pathway, where some of the protein itself will make the beta barrel in the outer membrane, and the rest of the protein comes out, and then it is excreted by a proteolysis, where the protein will be cut off and part of it will be delivered outside of the cell, whilst the rest of it is still making the channel here, the beta barrel. And this same protein that can also use an alternate, where it is not an autotransporter, but a single accessory factor, as we call it, or a single separate protein forms the beta barrel here and allows the protein, this complete protein, to be liberated into the environment. So definitely autotransporter is one way to do it, but then there is a single accessory factor, single accessory pathway that can be used also in this model. So easier for you to look at. The picture is much simpler, always in the white book that I usually find more appealing and easy to explain rather than that. But here it is. Your autotransporter, the pre-secretory protein, goes through the SEC system, and it has two domains in it. We call them the passenger domain and the helper domain. So the helper domain of the protein actually forms the channel in the outer membrane and allows the passenger domain to be exported out, where then it is, by proteolysis, will be released in the environment. Whilst the other way to look at it, using the chaperone-usher pathway, where the pre-secreted pilus subunit is being delivered into the periplasmic space, and a chaperone is used from the periplasmic space to guide it into the usher that is already preformed over there, and the growing end of the pilus eventually will allow the pilus to continue to extend out of the cell. So you can see that this is sometimes called the chaperone-usher pathway versus the auto-transporter pathway. Any questions? So here is a model where you can see which of the transport systems are dependent on the SEC system, which is found in your cytoplasmic membrane or the plasma membrane, and which of the secretory systems are SEC independent. So it's giving you a category to say that SEC dependent systems are type 5 and type 2, whilst your SEC independent systems are 1, 3, and 4. Does that help a little bit? This comes from a review in the Journal of Microbiology to give you a little more pictures of colored pictures of it, so that you see how these 5 systems that we've just described, how do they operate. And here's another one. You have a question?
Yeah, can you go back to these type 5 2 slides ago?
This one or before that?
So, oh no, the one that you were just at, sorry.
Okay, another one further, this one?
And so the first one, the autotransporter, that's the protein that is composed of like the helper domain and the passenger, so it transports itself out, basically.
On the outer membrane, yes.
And the one with the chaperone and usher, is the usher like a different protein unit?
Yeah. It is a different protein which is found, which is already embedded in your outer membrane. So it doesn't have anything to do with this protein that is being transported out.
Oh, okay.
And the example, of course, is your pilus protein is transported this way using a chaperone-usher pathway to get out or extend the growing end of the pilus occurs. Remember, the pilus is assembled at the base rather than at the tip. Your flagella is assembled at the tip, right? And this is where, when I showed you the pilus extrusion as well as when it retracts occurs because the growing end of the pilus is here. And we'll talk a little bit more as we give you more ideas. So the pilus production or extension of the pilus occurs through the chaperone-usher pathway, while some autotransporter proteins that I'm not giving you the names of because the people who will deliver it to you in the paper that is, so which autotransporter proteins, they will tell you about it and give you a lot of information on those autotransporters.
And I just wanted to make sure, these are 2 different systems. They're not like both type 5 or classified as type 5.
We are comparing the type 5 with the chaperone-usher systems. Okay.
Okay. Thanks, Dr. Rahman.
Anybody else? So here are more pictures from different books where you can compare type 1, 3, 2, and 4 in this one, and how these are some of the examples. And here is the one which is comparing all the 5 of them here. So you can just look at it once you get the understanding of each one of these as to what. And you can see the TAT pathway is also mentioned in this one. And in this one, the TAT pathway takes the folded proteins, remember, whilst the SEC takes the unfolded proteins. So definitely 2 different systems that are operating in your cytoplasmic membrane, whilst there are 5 different ones that connect the outer membrane to the cytoplasmic membrane and forming direct channels, or those that have been transported by the TAT or the SEC are being also channeled into these direct injectosomes, as we call them. Now, type 6 secretion system, I've just added a little more on it today. There is a 7 and an 8 already in your new version of your book. If you have the new version, you will see them, but I don't want you to get bogged down. But this is an interesting one in which there is a comparison of the bacteriophage. You remember the bacteriophage T4, which we studied in Bio 311 also, where the head and the tail structure of this T phenophages, we know that there is a core, the helical tail of the T4 is again contractile. So when it contracts, it actually allows the core to be driven through the whole cell wall of bacteria. Remember, bacteriophages have a challenge when they have to get through the rigid cell wall of a gram positive or gram negative, whichever. They have to go through that rigid cell wall. They can't go all the way in by themselves. What they have to do is to drive the core of the tail into the cell wall, and then they inject the DNA into the host cell. So this is an interesting way that we look at, where contraction of the helical tail of the phage allows the core to be driven in, and then DNA is injected. Whilst the similar system has been seen as a reverse, if you look at it, this is the type 6 secretion system, T6SSS, you could look at it, where the proteins from Vibrio cholerae and Salmonella typhimurium are being injected directly into the either another bacteria, or they could be injected into the host also. So examples of, this type 6 secretion system looks very similar to how the bacteriophage injects its DNA, but a reverse of it is seen, where the proteins that are assembled on the inside, they will contract and allow the core to be produced outwards. This is not the pilus, but the core will be projected out and will make contact with another cell out there. Okay. This in the eukaryotic system, if this is a eukaryotic cell, of course, virulence factors will be injected. But in some cases, bacterial symbiosis occurs, and this contact is made between two cells, so that some of the material from this cell will be going into the other one. Proteins will be secreted into the other one as a symbiotic factor. So this is an interesting one, where a lot of new papers have come out. Maybe if you're interested, you could read it, but this is contraction or contracted T6SS is compared to the contraction of the tail of the T4 phage or the T-even phages when it injects the DNA. Getting the gist of it? Yes, no? Questions? Yes, Anne, or did you have a question, or you're fine?
Oh, I'm starting to understand it a little more.
Very good, thank you. Okay, now I've gone up to 6. I'm not going to describe 7 and 8. Those are the new ones in the book, but just getting the idea as to how many secretion systems there are, and we try to compare them with whatever we know about the morphology or the physiology of the bacteria, so we can say that there is some commonalities and what are the differences in that case. So we talk a little bit about pathogenicity islands. So these pathogenicity islands, if you understand, they are not some island that you visit in your summer vacation or so, but these are large segments of DNA that carry virulence genes. So bacteria are kind of assembling those genes that help in the pathogenicity of this bacterium into a large segment of the DNA itself. So we kind of label those areas of the DNA as pathogenicity islands. And these, we have seen that we label them as pathogenicity islands because such islands are not present in the non-pathogenic variety of that bacterium. So there could be the non-pathogenic variety of that bacterium, no pathogenicity islands will be there. So it's a cute term that is being used, where some genes are assembled together that will contribute to the virulence or pathogenicity of that bacterium. This actually, we hypothesize that have been acquired during evolution in these pathogens by horizontal gene transfer. And we know that in a very good example of E. coli, which has become pathogenic, has taken over this plasmid from the cholera bacterium and has started to produce the toxin for itself. So by horizontal gene transfer between related bacteria may occur in nature and during evolution, and this way having those pathogenicity islands on the plasmids usually will be easy for the cells to exchange between them, so that a new bacterium will take on this pathogenic property. So as such, these pathogenicity islands also code for type 3 secretion systems also, so that the protein that is being made will also be transferred into the host system. So the genes for type 3 secretion system are located on the pathogenicity island. Now please note that instead of TTSS, we have gone back to P3SS, because now the type, since we have gone up to more of the system, we rather put them as numbers, T1, T2, T3 secretion systems, rather than, TT was originally for type 3 secretion system, but now we can call them T3SS or TTSS, whichever. So this type 3 secretion system enables your gram-negative bacteria to secrete or inject virulence proteins into the cytoplasm directly into the host, eukaryotic host. So it seems like this system is also used by many gram-negative bacteria that live in close association with the host. So those that are sitting in our colon or intestinal tract, they are the ones that may be injecting some of their proteins directly into the host system. But definitely this injectosome is a very important unit that is created by the T3 system, which will result in the ability to modulate the host cell activities also. Of course, it increases virulence, and these pathogenicity islands, as I said, of the stretches of DNA are not present in non-pathogenic members of the same genus of the species or the species. Now we come to the uptake of DNA and transfer systems in bacteria. So we know that there is exchange of DNA or transfer of DNA either from the environment into the cell by transformation, or from one cell to the other by conjugation. So what are the systems across the membrane that allow DNA to be transferred? And these have been studied extensively also, and definitely being looked at more closely as to what transfer systems are there in bacteria that allow DNA uptake. Now remember, DNA is a macromolecule, and to be able to go through membrane, there must be some system through which it will be taken in or be given out, right? So these DNA transfer systems we look as parallel to protein secretion system, and we have already identified some of our protein secretion systems that are capable of DNA transfer also. So in that same topic, we are now going to talk about DNA transfer and how it occurs. So if you look at transformation, that was first discovered by Griffith, transformation in Streptococcus pneumoniae, which is a gram-positive bacterium. This Streptococcus pneumoniae is naturally competent to take up the DNA from the environment, which has been released by some, cells that have lysed or they've opened up, and the DNA is excreted in the environment. And this DNA can now be taken up by a recipient cell. So the recipient cell should have a receptor where the DNA will bind. The fragment of the DNA and the whole large DNA pieces will not go in, but there's a limit to how big a piece of DNA that will go through in the process of transformation. So here there is an endonuclease that will be used, which will allow the DNA to be cut into smaller fragments first, because the large fragment cannot go through. So endonuclease will cut the DNA into smaller fragments. Then the recipient cell produces a transport system in its membrane where when the DNA is passing through it, it will degrade one of the strands of the DNA, and a single-stranded DNA will enter into the cell, which will be incorporated into the recipient DNA by homologous exchange here, and then eventually heteroduplex is formed, and it will be resolved into a DNA that is now has been repaired and has created the LAC plus gene. So you're looking at LAC plus gene coming into a LAC minus cell, and eventually the recipient becomes LAC plus. So this is the idea of transformation. generally as to DNA being transported across the recipient membrane by a channel, a secretion system, or an intake system, as you would call it. So looking at it, this has been really worked up quite well in a lot of papers. We won't be reading any of those, but to give you an idea that this transformation can occur in a gram-positive bacteria versus the gram-negative also. This is the gram-positive bacterium cell wall. You can see there's a thick peptidoglycan and the plasma membrane as their outer coverings. And here is your gram-negative bacteria, which has an outer membrane that has a peptidoglycan and has a plasma membrane. So the DNA has to go through in gram-negative and gram-positive through different layers of the envelope of the bacterium. I must mention that the term cell wall versus the cell envelope, you have to be very clear about. When I talk about cell envelope, the cell envelope will make up plasma membrane, the cell wall, even the capsule. These are all considered to be the cell envelope. Whilst the cell wall usually is limited to the peptidoglycan in your gram-positive bacteria, and the cell wall is always peptidoglycan plus the outer membrane. So this is the cell wall of gram-negative bacterium. This is the cell wall of gram-positive bacterium. If you want to include the plasma membrane, then you would have to call it the cell envelope. Please watch out for those words when you are giving me answers for the questions that I have asked in your previous homework. And even in the exam, see that you watch out for the terminology there. Okay, so the pill system, actually the system that is operating is the COM-E, which has been found in both gram-positive and gram-negative bacteria. A is the example of Neisseria gonorrhea, where the DNA is entering into, this is the gram-negative bacterium. Neisseria gonorrhea is a gram-negative bacterium, where the DNA has to enter through outer membrane, peptidoglycan, and plasma membrane, while the second example is Bacillus subtilis, which is your gram-positive bacteria, where the DNA has to go through only the peptidoglycan and the plasma membrane. So there are some elements that are common and some are different in these two, gram-positive and gram-negative. So the large protein machinery related to the protein secretion system involved in the DNA and the uptake of DNA occurs here. So the pill Q is an important one, which we will be transport across the outer membrane, which is not being found in your gram-positive because there is no outer membrane, so pill Q is not there. But the COM-E, A, and are there. There may be extra ones here, but remember the N is an important factor of your transport system, which is a nuclease. So the nuclease is associated with the transport system, which will degrade one strand of the double-stranded DNA that is entering into the cell. So the N is common. Again, that means in both gram-positive and gram-negative, a single-stranded DNA will be entering into the cytoplasm of the recipient cell, whilst the other proteins that are making a channel for the DNA to enter are the COM-E and COM-A and so on, right? Here's an FA, but you don't need to know all of that. Just be able to express what these pictures represent and how would you describe it to anybody. So we talk about conjugation. Now there are two cells that are conjugating together, and of course we call them donor and recipient. The donor and the recipient are usually differentiated by having, the donor will have the F plus plasmid. The fertility factor is called the F plasmid. So the cell that has the fertility factor, which is a plasmid, this is an extra chromosomal DNA, will allow this cell to become a donor. So this is the donor cell. And here is the recipient cell, which is considered to be an F minus. So the conjugation occurs between an F plus and F minus. So we name them as a donor cell, and this we will call the recipient cell. Now the donor cell is capable of producing a sex pilus, the F pilus as we call it. The F pilus gene is on the F factor. So this F factor is allowing the cell to act as a donor and to be able to either transfer its plasmid into the recipient, or in some cases also when this F factor is integrated into the chromosome, will also deliver some of the bacterial chromosome into the recipient cell. So there are two scenarios. We've talked about this in when we did our general microbiology course. The cross between F plus and F minus is being shown here. But if this was integrated, this would be called an HFR cell, high frequency of recombination cell, where now the plasmid will also drag some of the chromosomal DNA into the recipient. So in this case, we are just looking at the F plus F minus mating. And this F plus F minus mating will use a secretion system similar to type 4. So type 4, remember, is capable of delivering some DNA also. So this pilus contact first is established between the donor and the recipient, and the pilus, as I say, can retract. So retraction of the pilus is occurring so that the two donor and the recipient cells come in direct contact with each other. And now the type 4 secretion system is established, and this secretion system, type 4-like secretion system, will be established. And your DNA will elect the plasmid that has to go through this channel will be relaxed, because it is also a supercoiled DNA. So relaxosome will relax the DNA, and the coupling system allows remember the plasmid replicates by rolling circle model. Do you remember that rolling circle model? So when it is being duplicated by the rolling circle model, a single strand is created, and that single strand will enter through the coupling factor into the recipient cell. I'm making it a very simple explanation, but of course this is a lot of proteins involved in all of this transfer. But we are capitalizing on comparing different delivery systems. So we are just looking at type 4 secretion system being used for transfer of DNA between the donor and the recipient. And again, the single-stranded DNA will enter into the recipient. It will circularize, and again, using the DNA polymerase, will make itself into a double-stranded. And now both the donor and the recipient have the F factor, so both are called the F plus cells. So the result of F plus F minus mating in your gram-negative bacteria results in both becoming F plus cells. And now this recipient cell can act as a donor because it can synthesize its own pilus for future conjugations. Are you familiar with this whole concept of conjugation, or is this so new to you? Please ask questions now. Are you okay with this information?
Yeah, I think we're familiar with it.
Okay, so think about it, look at it. I don't want the details about conjugation. I want to only emphasize, remember, the type 4 secretion system. So the next slide will show you the secretion system encoded by the F factor. And that's what we are not describing every one of the protein. It is a very complex as such channel through which the DNA will be transported across. Just to give you an idea, that the sex pilus makes the initial contact. The pilus then retracts. The F plus cell, which is the donor cell, will prepare the type 4 secretion system. It's not the recipient that makes the secretion system, because the genes for the type 4 secretion system comes from the donor. The apparatus is laid, and a single strand of the copied DNA will be transferred into the recipient cell. And remember, it will need hydrolysis of some ATP. So this whole system of delivering DNA will use ATP from the donor. So in very simple terms is what I'm describing it. Are we okay? So different TRA proteins, TRA is always for transfer through conjugation. So the TRA proteins are definitely are coded all in the F factor of the donor. We move on to the last example for the day today is the Agrobacterium tumefaciens. This is again an example of type 4 secretion system. And you know that Agrobacterium tumefaciens, as was presented in our last student presentation, is a bacterium that is responsible for these outgrowths on the plant. And this is this Agrobacterium tumefacien is a alpha proteobacterium, and it causes crown gall disease in plants, right? So we're looking at the outgrowth due to the infection by the Agrobacterium into this plant. So this is a tumor-like growth that will occur in the plant where the Agrobacterium has infected the plant. And this genes for the plant infection is found on the TI, tumor-inducing plasmid, which is found in Agrobacterium tumefaciens. So the TI plasmid is the important plasmid which gives the ability to the Agrobacterium to cause this gall disease in plants. Okay? So what is this virulence genes in the Agrobacterium? So we found 21 virulence genes found in actually in 6 separate operons. So 6 operons are working to produce this 21 genes, the products of these 21 genes, which will be now expressed by this virulent form of Agrobacterium. Now remember, the Agrobacterium does not have to always be the cause of infection. It can live saprophytically in the soil and not have to do anything with the plants. So definitely these vir genes are not expressed. They may be present, but they are not expressed when the Agrobacterium is living saprophytically. But when it is in close association of plants, the plants are the one that will be inviting it to infect it, because they will be excreting or producing certain chemicals under cool acidic conditions that will bring the Agrobacterium closer to the plant and eventually cause the infection. So this infection occurs through a wound, not through through a very healthy plant. There has to be a wound where the Agrobacterium will infect, and it will activate the two, cause the activation of the 2-component signal transduction system. But the TI plasmid, the tDNA, is transferred from the bacterium to the plant, which looks like a type 4 secretion system. So type 4 secretion system example would be the Agrobacterium example, where the TI plasmid will be transferred into. So the tDNA directs the host to produce certain phytohormones, and these phytohormones will eventually cause the tumor production. I'm not interested in all of that, just that what kind of a transfer system is used to transfer the DNA from the bacterium into the plant. Okay, so this secretion system, where the plasmid, you can see in this figure, this is your Agrobacterium, where we'll be transferring the tDNA through the membranes into the host cell, and the tDNA integrates into the plant DNA and directs the synthesis of tumor, and also synthesizes certain opines. Opines are, of course, which are chemicals which are shown in this figure 2b. These chemicals will attract additional tumefacian cells and also serve as the carbon source for the infecting bacteria. So definitely transfer occurs between the bacterium and the eukaryotic plant cell, making a type 4 secretion system. Okay, we won't go into the details, but to give you a general idea as to how this happens and what secretion system is being used for TI plasmid to go from a bacterium into the plant cell. Well, that's a lot of information, but I hope you understand the focus was secretion systems and comparisons of them that you have to look at. So moving on, today we were supposed to do the chapter 3. Are there any questions on this chapter? We will get a little more information on some of these systems when we go to our next week's presentation. Students will present some review articles. So for these review articles, if you're writing a summary, you don't need to write the questions on it. Just make a summary of the paper. Okay, but you should produce some questions to address to the presenters, and you can gain your points when you ask questions to the opposite group. Okay, so remember, in this case, your summary will not have questions at the end. Anybody has any questions about this presentation? No? Then we are going to go ahead and share a new one, which will be a very common topic that you should be very happy to work on it and know about it. So I'm going to close this. So we share the screen now. And we go to okay. We talk about glycolysis. Now we are stepping into biochemistry part of it. Okay. Chemistry part of it, where a lot of reactions and structure will be involved, and this will be building upon what information you already have. So when we look at glycolysis, we look at it as a standard procedure in all bacteria. But then there are variations in different bacteria that we will emphasize. So I need to know that you understand what glycolysis is all about, and you remember the glycolytic pathway. Then only I can define some of the variations in different bacteria. How many of you know what is glycolysis? Anybody understands what the word glycolysis and the pathway which we call glycolysis is? Yes? Breakdown of glucose to make what? A 3-carbon compound? Pyruvate. So glycolysis goes from glucose oxidation to pyruvate. This is an oxidative process where glucose, a 6-carbon compound, will be converted to pyruvate, which is a 3-carbon compound, and then further breakdown of the pyruvate will occur. So what is the purpose of glucose oxidation, first of all? Why does the cell wants to break down the 6-carbon compound and bring it down to the 3-carbon compound? What is happening during glycolysis? It is making certain products that will be useful for other processes. Other than that, what could be the purpose of any oxidation?
Make ATP?
Yeah, to produce energy for the cell, right? So this is an energy producing pathway that we will talk about, right? So if you look at the summary of glycolysis, this is the summary, right? Glucose using up to ADPs, adenosine diphosphate, and 2 inorganic phosphates, and 2 NAD. We'll talk about what is NAD. And then it will go into two pyruvates, 2 3-carbon compounds. It will release 2 ATPs, and it will have donated some electrons to NAD, which is an electron carrier, and convert it from NAD to NADH plus H, right? This is how we write it. So remember, what are we getting out of glycolysis? 2 3-carbon compounds, 2 ATPs total, and some carriers that are now carrying the electrons that were taken or ripped away from the glucose, and they will be carrying it to the electron transport chain and be able to produce more energy for the cell, right? So the whole load down, do you understand the summary of glycolysis? So let's look at it as such. Oh, we've gone to the last one, so let's go ahead, go back to start with. Review. So some of the terms that you should know and you should work on it over the weekend or before the next class is what is metabolism, what is catabolism, anabolism, enzymes which are called holoenzymes, epoenzymes, cofactors, coenzymes, prosthetic groups, role of ATP,